I can start. Yeah.
Hi everyone. Hi everyone. Can you hear me all right?
Yep. We can. Yep.
Okay, great. And can you see my screen all right?
Yes.
Okay, great. So hi everyone. My name is James Roney, and today Sergey and I will be presenting on our work on scoring protein decoys with AlphaFold. So here's a brief overview of the section of the talk that I'm going to give. So first, I'm going to talk about a little bit of background on the protein folding problem, and then we'll talk about AlphaFold, which obviously is a recent protein folding method that's generated a lot of attention and a lot of excitement. Then we're going to talk about our new work on using AlphaFold for this application of decoy scoring. We'll talk about our specific findings there. I'm going to give a conclusion to this section, and then Sergey is going to talk about some other facets of our work. So first, I'll give a little background on the protein folding problem, and also feel free, if there's ever a pause, you can feel free to ask questions or ask questions at the end. Protein folding. So 3D protein structures are really important for building a mechanistic understanding of biology, for designing drugs, and just for really understanding how biology works at a molecular scale. And so a protein structure, 3D structure, is determined by its amino acid sequence. And we've known that since Anfinson's experiments, where basically you can take a protein, you can denature it so that it loses its fold, and then it can, when allowed to, it can just refold into its native structure. So basically, all of the information needed to determine this complex 3D structure is contained in just the linear primary sequence of amino acids. So more specifically, proteins are thought to fold to minimize free energy, which is a function of the protein's conformation. So this is often visualized as this potential energy landscape, where you have a few coordinates that represent sort of the conformational space of the protein, given by all the angles the backbone and the chains could form. And as you sort of traverse that space, the energy of the structure goes up and down. And physics will sort of drive that chain to fold into a structure that sort of forms a minimum in this energy landscape, and that's going to be the native structure. So theoretically, if we could understand this landscape well enough, we should be able to predict a protein structure from just its primary sequence. After all, this is how proteins fold in nature. So sort of a great goal to aspire to is to be able to sort of understand that potential landscape well enough that we can do a good job predicting protein structures from primary sequences. So there's this problem of predicting protein structures from sequences, is a really long-standing challenge. And recently, AlphaFold has demonstrated breakthrough performance on this task. And basically, the way AlphaFold works is it takes a couple of things as input. It takes sort of an amino acid sequence of a protein whose structure we want to predict. And then crucially, it also takes a multiple sequence alignment, which is basically an array of related protein sequences that are related to each other across evolution. So for instance, that's why this little graphic here from the AlphaFold paper has sort of different species represented on the vertical axis here. So each row would represent sort of a related protein from a different species, and each column would represent a position in that protein. And AlphaFold, as additional inputs, can also take some template protein structures, which are basically solved protein structures that are related in sequence to the target sequence. So basically, if you're trying to predict sequence A and sequence B has an experimentally resolved structure and is related through evolution and has some fairly high level of sequence identity, that structure might be sort of a good reference for AlphaFold to incorporate some of those features into its structure prediction. And so all of this information is then passed through a deep neural network with lots of fancy features. And the output is a 3D protein structure that we hope gives good agreement with the experiment, as is the case in this example. So as I mentioned before, those MSA inputs are really crucial for AlphaFold's performance. And so why is that? Well, a lot of recent progress in protein structure prediction has been driven by the use of coevolution information. So basically, if we have, so MSAs contain this coevolution signal, and it can be really useful for predicting protein structures. And the reason for that is basically we can look for positions, linearly distant positions in the MSA that show correlated patterns of mutation. And when that happens, that can be a signal that those residues are in contact in the folded protein structure. And intuitively, the reason that that might happen is if you have two residues that are in contact and one of them mutates, that then can induce a selective pressure for the second residue to mutate to compensate for that chemical change and sort of restore stability to the protein. And so what that looks like is then if you have natural selection acting on these proteins and sort of the sequences that you get in the MSA are those sequences that have been filtered by natural selection. What you see is that for residues that are in contact, when one position mutates, you might have sort of compensating correlated mutations that you see in the other position. And in this little toy example, like these two columns seem to sort of have correlated mutations. And so basically, if you then take all the positions and look at their covariance across that evolutionary space, positions with high covariance are can be treated as possible contacts in that folded structure. And basically all, or a large part of recent progress in protein structure prediction has been due to better utilizing this signal. And that's true for a lot of recent protein structure prediction models, including AlphaFold. So AlphaFold does a really good job predicting protein structures in a lot of cases, but without a deep enough MSA inputs, AlphaFold's prediction quality deteriorates. So you can see that sort of in this histogram, where we have a distribution of TM scores for predicted AlphaFold structures. TM score ranges from 0 to 1, 1 being a really good structure and sort of below 0.5 or 0.4 being a pretty poor prediction. And you can see that with the MSA inputs on this data set, AlphaFold does a pretty good job. Its TM scores are most often in the 0.9 to 1 range. But then when you just try to predict the structure without the MSA, then this performance deteriorates. And so this raises an interesting question, which is if the sequence of a protein contains all the necessary information to predict the structure, then why are our methods still so reliant on coevolution? And why can't we just learn to minimize the energy landscape over conformational space and arrive at the native structure? And so one intriguing possibility is that, so in theory, it should be possible to predict protein structures by minimizing that potential energy function over conformational space. But that is a really difficult optimization problem because there are so many possible conformations of a protein that searching them all or even searching a significant subspace would be really difficult and take a lot of intensive computation. And so one interesting hypothesis about how protein structure prediction like models like AlphaFold might work is that maybe they have learned an accurate potential function that can accurately assess candidate protein structures for their accuracy, but they still need an MSA to find a good starting point for this global optimization task. So even if you knew sort of a potential function that was somewhat akin to that potential energy, where the minimum corresponds to the native protein structure, it might still be really difficult to search over all of those structures. So maybe what models like AlphaFold do is they use these MSAs to provide a good initial guess at the structure. And then they can sort of perform a local search to refine that structure and arrive at a good prediction. And then AlphaFold and related models also have this added feature where they produce confidence outputs. And sort of in this hypothesized prediction workflow, you might expect these confidence outputs to correspond to basically the level of that internal potential function at the ultimate output structure. Okay, so now I'm going to talk about our experiments on decoy scoring, which sort of aims to sort of investigate whether this hypothesis is an accurate description of how AlphaFold works. So given this hypothesis, we don't really expect AlphaFold to be able to predict accurate structures without using an MSA, right? Because that conformational landscape is just so vast that it's difficult to find a good structure. So when AlphaFold is tasked with predicting the structure with just the from just the sequence, we get a structure that's not very accurate, and AlphaFold also knows that this structure is not very accurate, and its confidence metrics, as reported by the predicted LDT score and the predicted TM score, are pretty low. However, if AlphaFold has an internal potential function that's able to sort of discriminate good candidate structure from bad candidate structures, then we do expect that AlphaFold should be able to correctly assess protein structure quality without needing an MSA. And we can actually test that by taking advantage of AlphaFold's template mechanism. So basically, what we can do is we can supply AlphaFold with a decoy structure that's sort of like a proposal structure. So it's a structure that's not actually the native structure of the protein. In this case, this has a TM score of 0.56 to the native structure. And then we can supply that to AlphaFold as a template and see what happens. So what happens is AlphaFold produces an output structure that's pretty similar. It's slightly improved in TM score, and it also produces these confidence metrics. And so basically, given our hypothesis that AlphaFold sort of might have an internal potential function that's able to determine the accuracy of a candidate structure without using an MSA, we might wonder if AlphaFold's confidence metrics are actually a good predictor of the quality of this candidate structure, even when no MSA is provided. So basically, the question is whether AlphaFold can recognize a good structure when it sees one, even if it can't predict that structure from scratch. And so what we want to do to test this hypothesis is we want to use AlphaFold's outputs and its confidence outputs to see if we can actually predict the quality of that input structure without using any MSA inputs. And the way we did that was we computed a composite confidence, what we call a composite confidence score, by multiplying the predicted LDT score and the predicted TM score. And then we also have an additional correction term where we multiply by the TM score between this input structure and this output structure, which tends to be pretty high. In this case, it's 0.85. And the reason we do that is just because we're interested in scoring the quality of the decoy structure. And so if we have two decoy structures that end up giving us similar confidence outputs, we probably want to trust the one that changed less when AlphaFold sort of took it in and refined it. So that's what that extra little correction term does. And so, yeah, so the big question is, can AlphaFold recognize a good structure when it sees one, even if it can't predict that structure from scratch? And we found that the answer is actually yes. Even though AlphaFold can't predict these structures from scratch, its confidence signal, as measured by that composite confidence score I just mentioned, does an excellent job ranking confidence structures, ranking candidate structures, even with no MSA. So here's a plot where we took a bunch of decoy structures for a given protein. We fed them into AlphaFold, and then we said, okay, what does AlphaFold's confidence look like? And we actually see that as the decoy quality goes up, AlphaFold becomes more and more confident. And in this particular example, this red line indicates the quality of AlphaFold's prediction with no MSA and no template. So it's not like AlphaFold is just sort of comparing the decoy structure to what it would predict anyway. It's able to actually recognize when we get up here to the high TM score structures that these are high quality structures, even though it was unable to predict those structures from scratch. So I'll go into these findings in a little bit more detail now. So basically, we found that these rankings from AlphaFold work really well. So this is data from a decoy set that was curated by some people to create the Rosetta energy function, which is sort of a classical physics-inspired energy function that aims to sort of predict the stability of various protein structures. And we find that AlphaFold is able to, through this decoy ranking mechanism, outperform both the Rosetta energy function and deep accuracy net, which is a state-of-the-art ML model that was trained for this decoy discrimination task. And so 2 ways you can measure performance on this task are to measure sort of the correlation between this, this confidence score and the decoy score. And we find that AlphaFold has a better correlation than both Rosetta and deep accuracy net. And then another way is also just to look at the average quality of the top ranked TM score. And we also found that AlphaFold does really well in this metric. And then again, AlphaFold does a really good job ranking, even though it can't predict these structures from scratch with no MSA. This is also just another way of looking at the data. Here's a scatter plot of those top those top ranked TM scores, and AlphaFold is better at doing this than Rosetta and deep accuracy net on a strong majority of targets in this data set. And this also holds for sort of novel structures from the CASP-14 estimation of model accuracy test, where basically what they did was they took all of the top predictions for a given structure target from CASP, and then they had sort of additional models try to evaluate which of those predictions were best. And so AlphaFold does a pretty good job on this and outperforms all of the other sort of top performing models from that evaluation in CASP-14. So that's pretty interesting. And we also find that, consistent with our idea that AlphaFold might be sort of using this internal potential function to refine protein structures, we see that AlphaFold generally is able to improve on those decoy structures. In many cases, the structure improvement is somewhat modest, but in other cases, even with sort of a medium quality decoy, it's able to do a pretty good job and actually like hone in on the native structure. So that also is interesting and is consistent with this idea that AlphaFold might be sort of using MSAs and templates as a starting point and then doing some sort of geometry optimization from there. And so one detail here that I glossed over earlier is that if we take the default approach and just sort of paste the template structure, paste the native structure into the template mechanism, this doesn't actually work very well because AlphaFold is inclined to just copy over the template with high confidence. And that sort of makes sense, right? Because in AlphaFold's training, its job is just to get the structure right. And it was never trained with decoy or false templates. It was only trained with real templates. And so if AlphaFold sees a high sequence identity between the template structure and the structure it's trying to predict, it might just be inclined to copy that over and not really do any work and just say, okay, well, since this has such high sequence identity, I trust this structure a lot. And indeed, that's what happens if we just take the sort of naive approach of just putting the structure in as a template with that high sequence identity to the target structure. AlphaFold is just inclined to not really do much work, copy it over and say, okay, I'm pretty confident about this. So the way we actually, in our experiments, the way we deal with this overconfidence issue was to, instead of associating the decoy structure with the native sequence, we associated with the sequence of all gap tokens. And basically these gap tokens are sort of a token that exists in AlphaFold's sort of amino acid alphabet that's supposed to represent a missing or unknown amino acid. And so by doing this and also masking out all of the side chains, we actually then sort of force AlphaFold to sort of do its due diligence and actually sort of examine whether this template structure is something worth trusting. And when we do that, then we get really good correlation between AlphaFold's confidence and the quality of the decoy structure. I'll recap some of the stuff I just talked about and then give a few conclusions and further questions. So the takeaway here is that AlphaFold can score candidate protein structures very effectively, even without an MSA, and even though it fails to predict those structures from scratch. And this supports the idea that AlphaFold has learned some sort of internal potential function that needs MSAs and templates as a starting point for the global search problem of finding a good structure, but can still sort of assess the quality of candidate structures without being super dependent on MSA. And the code that we use to do this is available here if anyone wants to look at it. And so this idea that AlphaFold might sort of be using might sort of have this internal potential function has also been supported and applied in a recent paper from the Baker group, where basically they used a similar approach to what we did to sort of give AlphaFolds like designed protein-protein interactions that they created using Rosetta as sort of a template, and see how confident AlphaFold was in sort of the accuracy of that predicted protein-protein interaction. And when they did that and filtered for designed interactions that had high AlphaFold confidence, they were able to really increase the experimental success rate of their binder design process, which sort of is consistent with our results that AlphaFold can do this sort of decoy discrimination task pretty well. And so there are still some questions left open by this, though. So one is, well, does AlphaFold's learned potential function reflect protein physics? Like because that's sort of, I guess, a big goal of sort of the next phase of our journey to understand protein folding through machine learning is, to actually sort of have models that are more in line with the physics that drives protein folding. And so I think there's some preliminary evidence that the answer might be yes, because, we can do this discrimination well without MSAs. And also, this recent application to protein binder design suggests that it can work in a space even when, you're looking at sort of interactions that are definitely not something that are pretty different from things that we've seen in the training data. But I think there's still more investigation needed to sort of really investigate whether that's the case or not. And then another question that's been opened up by recent research is how AlphaFold's single sequence decoy discrimination ability compares to the abilities of language model-based single sequence models. And these are like OmegaFold and various other models that sort of don't use MSAs, but instead sort of substitute that for the evolutionary information learned by language models. And so if we think that AlphaFold's sort of internal potential function to sort of understand something about protein physics, then we might expect that we can actually go beyond the abilities of these language model-based approaches, and maybe even predict structures, predict or refine structures, or successfully rank decoys for structures where coevolution or language-based models fail. But that's sort of a direction for further research. Another thing is if we want to rank protein structures without coevolution information, how do we deal with sequences that might be oligomers? Because if we're basically striving for a sort of physics-based or physics-inspired understanding of protein folding, it might be difficult to understand, how actually multiple copies of a protein are supposed to sort of interact together to form a stable structure. And lastly, so we've talked about how AlphaFold, even without an MSA input, can do a good job discriminating decoy structures, but I haven't yet talked about how we might want to apply that to the problem of structure prediction from scratch, because we looked at data sets where we already had a lot of decoys generated, and we could use AlphaFold to discriminate those decoys. But in theory, we actually want to be able to predict new protein structures without necessarily having that decoy library at our disposal. And so I think now Sergey is going to talk about some approaches to sort of extend these results to predicting new protein structures. Yeah, thank you. And I'm not sure if we want to do some questions now, or we could wait until after Sergey's portion.
I think if anybody has questions, we should start now, just because, well, what James just presented is fresh on your mind. If anyone has questions, feel free to speak up.
Hi, I have a quick question. By the way, James, great job with the presentation. I really liked it.
Thank you.
But I want to, yeah, no worries. I just want to ask, so how do you guys actually go about creating each decoy structure? Is there a specific method that you guys use to create like, I guess like a decoy structure for some kind of new protein that's never been seen before or something like that?
Yeah. So what we were doing was we've used existing data sets for these experiments that I've just presented. So the Rosetta decoy data set, basically what they did to make that was they used a bunch of different protein structure prediction methods, I think. So they did things like perturbing the native structure, use homology modeling, try to relax a bunch of structures using potential functions like Rosetta, and things like that. The CASP data set, that's basically a bunch of different protein structure prediction servers that were entered in CASP trying to predict these structures. And as they try to predict those structures, we sort of naturally generate a library of less than perfect decoy structures that you can try to rank. And so, yeah, so generally the way these decoy data sets are generated is just they throw a bunch of structure prediction methods at these targets and see what they come up with, and also potentially take the native structure and perturb it and things like that. But actually, in the next part of the presentation, Sergey, I think, is going to talk about how we can sort of bootstrap that process a little bit.
Yeah, cool. Thank you very much for answering my question.
Yeah, sure.
Hey, I have a quick question as well. You mentioned you introduce little gaps in the decoy structure so as to force the model not to have that one-to-one correspondence between input and output.
Yeah.
Is that akin to like, how you kind of like in a VAE, you kind of introduce Gaussian noise in the inputs or the outputs? Is that kind of like the same thing you're doing?
No, I think it's a little bit simpler than that. Basically, the way maybe there's a slide that would be helpful for this. So maybe basically the way the template is featureized. Can you see this? Do you still see this slide?
Yeah.
Yeah, the way the template is featureized, basically, the template is inputted into AlphaFold. Basically, it's you take a distance matrix, so the distance between all pairs of residues, and that gets passed into AlphaFold. And also the things that also get passed are a series of torsion angles from the side chains in the backbone, and also basically a one-hot encoded amino acid sequence. And the reason the one-hot encoded amino acid sequence is important is because AlphaFold has probably learned to look at that sequence and use it to decide whether to trust this template very much or not. Right. So in training, AlphaFold was trained with a wide variety of template qualities. They actually did some sort of data augmentation procedure where they don't always give it the best template. Sometimes they sort of reshuffle them so that AlphaFold is forced to sometimes contend with templates that aren't very good. And I mean, in homology modeling, right, a pretty good predictor of how good the protein template is going to be is how much sequence identity you have between the protein you're trying to predict and the template you have at hand, right? And so because that was how AlphaFold was trained, it probably, when it sees a template with really high sequence identity to the thing that it's trying to predict, it's probably learned to say, oh, okay, well, that's probably a pretty good structure, which is completely rational, right? Especially if the sequence identity is 100%, right? It's basically bound to be perfect, right? So what we just did was we took that one hot encoded amino acid sequence associated with the template and just set it all to the gap token to try to make that not happen. And we found that works.
Okay, got it. Thank you.
Great. Any other questions? All right. Well, thanks, James, so much for the talk. So I'm just here to supplement some of the things that James said and some of the additional experiments that we've done. But if anyone has any questions, feel free to interrupt. So let me begin. So one thing I wanted to show is that recently I tried to re-implement some of the things James did within our Colab design notebook. And the reason for that was because I was trying to interface it with some design procedures. But I also realized it might be helpful in some regards for people who just want to try to do these kind of things on Google Colab instead of on their local computer. So I thought I might share that first, just in case anybody's interested in running it on their own data set. And if anybody does run it on their own data set, please share with us what you got as your results. I think it's always really exciting to see what happens and how these tools are used. Even if you decide you're not going to publish that data, I think we'd still be curious to see what results you got and how the tool has been useful for you. Okay, so I'll start by sharing my screen. So to begin, we have a Colab notebook, which I'm calling AF2 rank notebook. And this notebook is similar to the protocol that James described. There is one minor difference, and that minor difference doesn't seem to make a huge difference, where in the default AlphaFold protocol, 15% of the MSA features are randomly masked, while in this notebook, because we were specifically targeting it for protein design, we don't want to mask anything at all. We just want to give it the actual sequence. And so that's one difference. So this notebook I'm about to show you won't perfectly replicate the results, but it's actually almost like 99% correlated. It's just that 0.1% it's not correlated. That's where it appears to be coming from, the difference. And so I set up this notebook here. And so by default, what we'll do is it'll download those exact data sets that we talked about. And so if you go through this notebook, you can decide you want to do some experiments. And these are some of the experiments that James talked about. We say, hey, don't replace the sequence, replace the sequence with gap. But also you could also try out some things like replace with alanine character. You could technically like click in here and type in your own character if you want. And the other thing is to mask or not mask the side chains, how many recycles to use, which model to use. And one other thing that we're kind of excited about is the fact that other people have now been releasing their own model weights. So besides just AlphaFold weights, you can now have like OpenFold weights. Though towards the end, I'll show you some experiments that I've done with the OpenFold weights. It turns out that they don't appear to work as well. And we're still kind of trying to debug why that is. And we think it's because it's sort of not actually paying attention to the template sequence. But in short, you can specify your options. You can specify your native structure, your decoy set, and it will create a model, set the wild type sequence, and then it will just march through and score all the things. And then later on, you can make plots like the ones that James showed, just to show you that this actually works. Yeah, you have to trust whatever code you're running is real. And so then you could start going through and start scoring your decoys. One thing I wanted to show here is that it's relatively fast. And so here you could see the input template. In this case, the first template is just actually the native structure, the output TM score, the TM score between the input and the output, and then the composite score that James talked about. But also we have the predicted TM score, PLDDT. And so here you could kind of see, like, for example, like a bad template like these guys here. Oh, shoot, you know what? I think I might have forgotten to run this cell. Let me rerun that 1 second. So you could see, like, for example, templates that are bad have a bad predicted TM score, and so on. But now and then later on, you can go in here, for example, plot what that looks like. And so this case, you see the correlation. But in this case, let's say if you decide, you know what, I'm not going to hide that sequence. I'm just going to use the original sequence that is actually inside the template. And then if you start running this, you'll now see that this correlation that James talked about starts to go away now, because like all these templates that are even bad are now having pretty much the same predicted TM score. And so if you plot that, you can see what's going on here. See, now it's just not correlated at all. So if you wanted to play around with that features, it's all in this notebook here. And then you could also plot like TM scores, predict TM scores, see how you're improving over the input structure and stuff like that. Okay, so that's the notebook, and it's part of Colab design right now. And so if you have a data set and you want to try scoring it pretty quickly, it's now present here. Oh, and if you do use it, don't forget to cite us. Okay, so now the question is like, okay, now we know that we can. Oh, by the way, before I go on, does anybody have any questions about the notebook or anything I just said? Concerns? All right, I will just keep going then. So one thing that this idea that we can discriminate templates and we can rose to us is that, can we now potentially use this to generate decoys? And so what I mean by that is you have, for example, an AlphaFold instance here that takes in some sequence, and this could be literally any random sequence, and it outputs a structure. And literally anything you put in here will always give you back a structure. And now we could take the structure and potentially feed it into another instance of AlphaFold as a template, and then you can use that potentially get your confidence and then backpropagate that signal, update this random vector, and just keep looping this around. And so we were kind of curious, would something like this work? Like, could we actually use an instance of AlphaFold as a generator and another instance of AlphaFold as a discriminator? And because but just to give you a bit more details, here's a slightly simplified version of that slide, where the idea is like you could make random mutations to sequence, have a generator output a structure or a distance matrix. In this case, because the template is literally just a distance histogram input, you can then literally feed the predicted histogram into the discriminator model, and then you can compute some confidence based on the outputs of the model, and then just keep looping this around. And here's an animation of that in action. So in this case, we literally start with the wild type sequence, which initially is mispredicted. And 1 second, this animation will restart in 1 second. And because it's a GIF, it's a little hard to, okay, here we go. So it starts off by the correct sequence. And then what we do is we just keep randomly updating this sequence and watch, for example, the TM score get better and our loss function, which is our confidence, get lower. So in this case, it's the inverse of the confidence. And so in this particular example, starting with some random structure, which turns out it's actually not that far from the correct structure, you could sort of move around and sample some input vector that gives you back the correct structure. And so this sort of got us really excited, and we wanted to see whether or not this works for other examples. But it turns out that it doesn't always work. In some cases, you quickly find the correct solution, but a lot of cases you sort of get stuck and you don't really get really far. And in some cases there, like in this particular case, you sort of found this alternative minima and you kind of got stuck in there. Now, there is a possibility, let's say if we did a huge amount of sampling, that, let's say, the correct solution would even have a better score. And we believe that that's going to be true because we kind of see that already with these pre-generated decoy sets. But at least just using this really simple idea of, say, linking up an instance of AlphaFold as a generator and discriminator, this procedure, starting with the wild type sequence here, sometimes has you get stuck in some local minimas. But just to show you overall, now if we just compare this, for example, to a standard AlphaFold run where we have our model and then we just do recycling, what we see is the following. So this is, on the x-axis is the standard AlphaFold protocol that only uses a single sequence and just does 3 rounds of recycle, versus this I don't know, alpha GAN-like model. And if you just keep optimizing this mutant sequence, what we find is often we can find a better structure than the default protocol. But there's a whole bunch of examples where it doesn't really get better. But one promising thing is that these guys that don't get better tend to also be less confident, meaning that the reason why I didn't get better is not because there's an issue with the energy function, but because it might be just a sampling issue. And so one future direction that we're really excited about is, can we somehow improve this generator? This could either be a neural network or maybe just even using Rosetta to generate more decoys, like sort of being supplemented by the AlphaFold score function, or maybe there's some other generator that somebody trained that could potentially be used in these regards. And so we're pretty excited about potentially doing a better job. And I think there's already a pretty good signal. And one other thing, as I said, for those of you guys that are here on this call, the Open BioML people, if you guys are interested in this kind of project and want to potentially help out, I think this is an area that we're really excited to take further, but it would require a lot of computational resources. And I think if people here are really excited, we can potentially work on some of these projects together to see if we can improve these models. So now you might wonder, okay, we showed, so James already showed you guys that we can rank structures really well for a given sequence. But now the question is, can we do the opposite? Like, can we actually rank sequences for a given structure? And one way to potentially answer this question is to sort of formulate this question as sort of a design problem. And what I mean by that is, imagine we take a structure with a given sequence, we remove that sequence, and then we use some algorithm. In this case, we can use AlphaFold to find a new sequence that it likes, and then we can compare the sequence from the original structure to the sequence that is generated, and see how close they are to each other. And so the idea here is that if AlphaFold, for example, it has a really good understanding of mapping of structure to sequence, then using this procedure where you actually input the structure as input, it should result in a higher sequence recovery. And so just to summarize this, the idea here is like you could take a structure, extract, for example, the distogram from it, which is just the pairwise distances from every pair of residues. You can start with some random sequence, pass it to AlphaFold, and make a prediction. And then what you could do is you could say, okay, I'm going to look at the predicted output from AlphaFold, and then my target output that represents a structure, compute some loss, and then back propagate that loss to get a gradient, and then apply that gradient to update my sequence, and just keep looping this around. But one thing that we're now going to change is we're going to say, well, let's also take this target, like this, what we actually want, and pass it in as input to AlphaFold. Now, one thing like the expectation, like the first time I thought of doing this, our expectation was that it would actually make things worse, because like if you're feeding it the target, then it's going to output the target, and there's going to be no incentive to improve the sequence. Like if AlphaFold can just predict the structure because it just passes the structure right through, then you would expect that this method, like this procedure of protein design, would actually make the sequence worse. But what we found was actually quite the opposite. Just to contrast the two ideas here, one is we're designing with template input, and one we're designing with no template input, like only the loss, but this is just going a single sequence in. And so here's the results. So this is no template, just doing design and trying to match the RMSD of a given structure, and with template input. And the idea is here, when you added the template as an input, we actually saw that the sequence recovery improved, which was actually the opposite of what I expected, and I was quite surprised by this. And because I was actually I assumed everything would just get worse, because as soon as you give the template, there's like no incentive to do anything. But it turns out that it does actually improve things, and that's the case both for this monomeric. This is the Rosetta dataset that James talked about, but also I just wanted to confirm that there wasn't some overfitting going on, and I also used the free modeling targets from Cas14. So these are proteins that a lot of groups didn't do so well, and so we kind of assumed that these are proteins that are really, really different from anything in the prior training sets. And so if anybody wants to actually try this out, I think one of the exciting things about this is that if, let's say, for example, you want to design a protein for a structure that's really, really complex, and AlphaFold just cannot predict without an MSA, you can use a template as a substitute. But the key here is the template is not going to have a sequence. The template is just going to be all gaps, all the side chains masked, so there's no way for it to cheat. And so that could be a way to potentially bypass that issue related to MSA. And so we just wanted to mention that we do have a collab design notebook, and you guys may have seen me talk about this. And so one feature that we added to this is this idea, say, okay, when you do fixed backbone design for a given PDB file, you could specify the template input. But the other thing we did was, just to make sure you don't accidentally overfit on this template, is to also introduce some kind of dropout. So what we're doing is the template, like 15% of the positions of the template are dropped out randomly during the design procedure. And so then if you just run this procedure, you start off with some random sequence, and that random sequence actually has a really, really high RMSD. And as you keep changing the sequence over rounds of iteration, in this case, I'm cycling between the two different models, the two AlphaFold models, and you could see the sequence, the RMSD starts to drop even after just a few steps, but the sequence recovery just continues to climb, even though the RMSD appears to be the same. But the reason why it continues to climb is because if you actually look at the confidence now, for example, now if you just stare at the confidence, so even though the RMSD has dropped, the confidence values are still actually improving over time. And because of that reason, you start to see that the sequence recovery starts to go up. Now the sequence recovery is not as high as some of those fancy new MPNN algorithms out there, but it gives us a lot of evidence now that there is indeed, the model has indeed learned a function that maps sequence to structure. Because at the very beginning of the optimization, you could see here, I'm literally giving it the template information and it cannot predict it. And it's only once you find a good sequence, in this case, a good sequence with high sequence recovery, then that it starts to recognize, okay, this is actually a good structure. I'm going to give you a good RMSD and a high confidence output. So here's this is plot. This is the RMSD, you can see is dropping and the sequence recovery starts to go up. Here's what the actual structure looks like. If you want to take a look at it, you can see it's actually, I would say it doesn't look that terrible. I guess people who do protein design can tell me otherwise, but you can see like there's hydrophobics inside and all that kind of stuff. I guess one thing I'd say is that color by HP. You can sort of say, look where the hydrophobic residues are. So in this case, it looks like it puts some hydrophobics on the outside, which might be a bad thing because it could potentially form a homodimer. I guess we could try animate this to see like the two structures that it predicted. And so these are the two predicted structures coming from model 0 and 1. And finally, we could look at the actual trajectory here. So we're starting from the actual, at the very beginning of the trial, the sequence is literally all zeros, and we start running it. Then it finds some representation of the sequence, and it's very, very confident. Towards the end, we actually end up getting, as the temperature goes down, we actually get one hot sequence at the end. And then this is your sequence here. And so this idea of using templates is now implemented inside our Colab design notebook. Any questions or concerns? Just make sure that everybody's still on here. Okay.
Sorry, would you mind if I ask a quick question?
Oh, go ahead, of course.
Did you guys have the opportunity to test any of the structures yet?
By test, do you mean like express them or?
Yeah, like synthesize the genes and after express them in like E. coli or yeast or something.
That's a good question. So we haven't, but other people have used AlphaFold for design and did test them. But so far, like these sequences haven't been tested, the ones where we use template inputs. So we know that the sequence recovery improves, and so the assumption is that the structure, I mean, the sequences you get back are going to be better. But we haven't experimentally tested these.
Okay, yeah, that makes sense.
Yeah,
plus it's pretty new too.
Yeah, I mean, also like here we weren't really trying to suggest, oh, this is a new protein design method. Here it was more like we wanted just to demonstrate that it looks like AlphaFold did learn some kind of sequence structure relationship. But it's also possible, like for example, the reason why this is at 30 sequence recovery on average and not like 50 is because it's possible, like as soon as it finds a few key residues that match the structure really well, it might have not any incentive to go further. And so we don't expect it to be a perfect function. So you probably still want to supplement it with other energy functions as well.
Yeah, that makes sense. One thing that I was thinking about when you were going through the presentation was it seems like a pretty good way of doing in silico mutagenesis.
Yep. That's a good segue into potentially a bonus feature that I added to this notebook. So if you are curious in doing some in silico mutations, I also set up this little example here where, so this is actually an example from Mike Lob's data set from our collaboration many years ago, where what they did was they actually took a protein pair. And so this is a toxin, antitoxin protein pair. And they identified a few key residues that were previously shown to be covariant between the two interfaces. And they actually tried every possible mutation, like they mutated every combination of these 4 amino acids. And then the cool thing about toxin, antitoxin is like the readout is literally like, does your organism live or die? And so you could quickly do a bunch of screens and then literally just select for alive and dead animals, I guess. But well, in this case, it's an E. coli, so it's not really an animal. But the cool thing is we have this data set, and I just had it laying around from my former collaboration. And this is actually published. Unfortunately I forgot to include the reference here. And so I was kind of curious how well will it actually do on ranking mutations. Actually, in the paper, James also looked at I believe beta lactamase is another data set. So and that's in the paper. But I think in this example, this is something I just pulled out for this tutorial. And so here we have the two proteins, the first protein and the second protein. And this is just the files that have those data set included. And so here what you could do is in the same notebook, this is also using the Colab design notebook, Colab design algorithm. What you could do is you could say, I'm gonna do fixed backbone design. So in this case, we're not actually going to do design, we're going to rank sequences. Tell it that we're going to use templates. This template, and there are two chains here. This protein has two chains in it. And then what we're going to do is we're going to set the sequence to wild type. And we're going to set the number of recycles to do. And then what this little for loop does here is literally just loops through the data set of mutants and the sequence, and then creates this mutant sequence, tells the model to predict that sequence, and then tabulates all the different scores. In this case, like TM in output, RMSD input output, composite, and so on. And this is just a quick test that I did. I haven't really tried to do anything better. This is just something in particular for this tutorial. And what we could see here is that it's not perfectly correlated, but the cool thing is, so this is fitness. And what Mike Lob's people tell me is about 0.1 usually means that you're pretty fit. And so what we see here is that it appears that all the guys that are fit, AlphaFold actually likes a lot. But things that are less fit, in this case, there are some things that are, I guess you could say, false positives here. And I think this is sort of consistent. What people have been seeing is that it turns out like AlphaFold can predict all de novo designed proteins, but then there's a whole bunch of other proteins that AlphaFold can also predict but don't work. So it looks like it's a really good positive filter, but maybe not a good negative filter. I don't know if that's the right way to say it. So the idea here is like if AlphaFold likes your protein, it might work, but if your AlphaFold does not like your protein, it's probably most likely will not work. So it could probably be used to screen out all the potential negatives. So let's say if you just cut off all your designs here, this is all the how much human resources or pipetting, whatever you can save money on. But of course, there are going to be a bunch of false positives here, as you can see here. And so, yeah, it does look like you could potentially do in silico mutagenesis, but it's not a perfect correlation. And we've also only looked at like 2 different data sets, so it's possible maybe for other selection scheme there's going to be a stronger signal. The other thing I haven't done, I don't know if James looked at this yet, but maybe you could do something smarter, or like instead of taking like the average composite score, which is probably useful for ranking decoys, maybe you could potentially look at per position confidence levels, because if you're just mutating one position, maybe you care more about the confidence of that position. So that's something I haven't looked for this data set. But I just wanted to point out that the functionality to do this is actually already implemented, and you could just start going through mutating your sequence. And this is actually, from what I remember, so this is just starting from a new session just to show you how fast this goes. And so one thing about AlphaFold is that the first time you run it, it actually takes, I believe, like 20 to 30 seconds to compile. So this is the way the first time we're running it with this first mutant sequence, we'll have to wait a little bit. It's going to take 18, 19, 20. Well, actually, while this is running, does anybody have any other questions?
It's more of a comment. I was gonna say that one thing that I plan on like maybe checking out is taking like sequences that are either generated from like protein MPN or maybe even this, and after using other models like ProGen that provide some kind of scoring function to kind of like rank those predictions. Or even like the ESM models, for example, ESM 1V and 1F, I believe both of them provide options for scoring structures. So I could be wrong about ESM 1F, but or I mean, sorry, 1V. But yeah, regardless, you could probably create like an ensemble of different models to kind of score different sequences as a way of I guess screening for some of those sequences that AlphaFold might not catch.
Yeah. I think that definitely makes sense, because I mean, one way you could think about this is that what AlphaFold effectively is modeling is the probability of structure given sequence. And sorry, I'm writing with my mouse. This is a little ugly. So this is what AlphaFold is modeling. But the one thing we should keep in mind is that AlphaFold is only trained with valid sequences. So it assumes that the sequence you give it is a valid sequence. And the reason why this is critical is because you could imagine if you're training a model with a limited number of parameters, and the model will probably find the most important residues that it thinks is important for that structure, like to be able to predict that structure. It might just potentially ignore everything else. So in order to sort of model this joint probability, you probably also need to include like the probability of sequence, which effectively is what the language model is. The language model is modeling this. You can think of the AlphaFold as modeling this, and then methods like protein and PNN, and then models like the ESM iFold, for example. Inverse folding is what they call it. It's actually modeling the probability of sequence given structure. So I guess what I'm trying to say is that even theoretically, it doesn't make sense to only use this, because this assumes that the sequence is valid. It also doesn't make any sense to use this because this assumes that this structure won't change. Because in what might actually happen is like you might actually get a scenario where you think you found a sequence that matched that structure really well, but then that sequence might actually fold into a completely different structure and might have even a lower free energy. And then it might not make a lot of sense to only use this either, because it's possible that you start with some sequence and you start to move away, and then if you're not sort of conditioning on structure, you start to move away in some other structure space. So like the idea is like if you use language model for design, there's a possibility you found a better pseudo perplexity or mask language modeling loss, but now you've actually coding for a completely different structure. And so I think at the end of the day, you probably want to combine all of these models together to really truly model the probability of structure sequence and structure. So I guess what I would say is if you are going to use these methods for design, I would highly recommend trying to figure out a way to combine all of them, because none of these models alone is actually giving you the true probability, joint probability distribution.
Yeah, I agree with that. I think you're absolutely right.
Thanks. Okay, so I think that might have been the only thing I wanted to say for now. So yeah, so you can see here now it's running through and doing the design, the scoring of the sequences process here. And I think this data set has like 10,000 or so examples. So this is going to take quite some time, but you can see it's actually running relatively fast. I mean, you might have to set this up and then go get some coffee and come back for this. Okay, so I think that was the main thing I wanted to say. Oh shoot, let me clear my screen. Does anybody have any? Oh, I guess I guess thank people in my lab. All of our code is available. Check it out. Some of my funding sources. And I guess I'll maybe take a few more questions if there aren't any. Or concerns you want to voice?
Maybe I can add a general comment about. I absolutely loved the addition of the Colab notebook that during the presentation. The interactive part in particular was great. So thank you for that. The presentation in general was amazing. And for those, maybe I can add as well another thing related to the community more generally, in that for people, as Sergey mentioned, there might be opportunity for collaboration in terms of at the community level for a project related to these topics. And even more generally, we might have or we will have most likely a project soon intended for models related to AlphaFold. So I'm hopeful we'll be able to share more soon. All right, I think if there are no more questions, I will advance a big thank you to both James and Sergey for the presentation and for joining us today for the Journal Club. Lot of applauses in the emoticons and the reactions. All right, I think we can maybe stop the recording and see you folks next time then.
Mhm.
All right, thank you so much.